Hi, yeah, my name is Sattva Neelapu. I'm professor and deputy chair at MD Anderson Cancer Center in Houston, Texas. Today, I'll be discussing with you the phase 1 interim results from the ALPHA study, which tested ALLO-501 and ALLO-647 in patients with relapsed or refractory large B-cell lymphoma and follicular lymphoma. So the ALPHA study is a phase 1 open-label multicenter study being conducted at 6 centers across the United States. This tested a novel product called ALLO-501, which is allogeneic CAR T-cell therapy. To be successful allogeneic CAR T-cell therapy, we need to be able to prevent graft-versus-host disease and also prevent or delay the rejection of the donor CAR T-cells to allow sufficient CAR T-cell expansion and induction of anti-tumor T-cell responses. So in the case of ALLO-501, the graft-versus-host disease is prevented by knocking out T-cell receptor using TALEN gene editing technology. And to delay the rejection, the CD52 is knocked out to allow the usage of ALLO-647, which is a CD52 monoclonal antibody, for conditioning the patient to induce host T-cell depletion and allow CAR T-cell expansion and induction of anti-tumor responses. So in this phase 1 study, there were a total of 22 patients who've been enrolled and treated to date. At the time of the data cutoff for the ASCO presentation, 19 patients were evaluable for efficacy, and all 22 patients were evaluable for safety. We tested 3 dose levels of the ALLO-501 at a flat dose of 40 million, 120 million, and 360 million. And 2 and 3 different conditioning regimens were also tested, where patients received cyclophosphamide and fludarabine along with a low dose or a high dose ALLO-647 monoclonal antibody, either in a concomitantly with Cy/Flu or in a staggered manner. To be eligible, patients had to have either relapsed or refractory large B-cell lymphoma or follicular lymphoma after at least 2 lines of therapy. The therapy has been fairly well tolerated. To date, we have not observed any dose-limiting toxicity, graft-versus-host disease, or neurological toxicity, also called as ICANS. A third of the patients developed cytokine release syndrome. Majority of these were grade 1 and grade 2, with only 1 patient having grade 3 cytokine release syndrome, which was reversible within 48 hours. About half the patients had infusion-related reactions to the ALLO-647 monoclonal antibody, but these were also reversible, and there were no dose interruptions for any of the patients. 19 patients were evaluable for efficacy. The best overall response rate to date was 63%, and the best complete response rate was 37%. And responses were observed at all dose levels tested. The higher dose of ALLO-647 conditioning allowed a deeper lymphodepletion in the host, as well as a more delayed T-cell recovery, and was associated with a higher complete response rate of 50% versus 27% on the lower ALLO-647 conditioning regimen. And the responses were associated with CAR T-cell expansion, with patients achieving a complete remission or partial remission having a higher CAR T-cell expansion versus those who had only stable disease or progressive disease. So overall, I think this study to date shows that this combination of ALLO-647 and ALLO-501 is fairly well tolerated. There's no dose-limiting toxicity of graft-versus-host disease observed to date, nor were there any neurological toxicities observed. Cytokine release syndrome was observed in only a third of the patients. It was largely manageable. And the best overall response rate and complete response rates appear to be comparable to what's been observed with autologous CAR T-cell therapy in lymphoma patients. And at the time, at a median follow-up of 4 months at the time of the study presentation at ASCO, 75% of the responses were ongoing. So currently, dose escalation is still ongoing on the study, for both the conditioning regimen as well as the ALLO-501 dose. And a new study called Alpha-2 study has also been initiated, where the rituximab safety switch was removed from the ALLO-501 CAR-T product.